Most animals age for one simple reason: cells degrade faster than they can repair themselves. Every heartbeat, every breath produces free radicals, reactive molecules that damage DNA, proteins, and mitochondria. With time, these microscopic injuries accumulate. Telomeres, the protective caps on our chromosomes, shorten with every cell division. When they become too short, the cell stops dividing, enters senescence, or dies. This constant erosion of cellular integrity is what we call aging. But not all creatures obey this rule. Some slow aging to a crawl, some resist it, and one animal may have escaped it entirely. In the remote Galapagos Islands, life moves at a different pace. Here, volcanic rock meets endless ocean, and evolution experiments in slow motion. Among these ancient landscapes lives a creature that seems to defy time itself. Giant tortoises, living relics of an older Earth, and among the longest-lived vertebrates ever recorded. Giant tortoises can live more than 150 years, some of the longest-lived vertebrates on Earth. Their secret begins with metabolism. Tortoises operate on extremely low metabolic rates. They burn energy slowly, produce fewer free radicals, and generate far less oxidative stress than most animals their size. Their heart beats far slower than mammals, and their cells divide more cautiously, reducing the chance of cancer-causing mutations. Recent genetic studies have revealed even deeper clues. Giant tortoises possess enhanced versions of DNA repair genes and tumor suppressor genes, molecular guardians that patrol the genome. Among them are extra copies of TP53, a gene often called the guardian of the genome, responsible for stopping damaged cells before they become cancerous. This expanded network of protective genes, combined with their slow, deliberate metabolism, creates a body that ages at a crawl, a system built not for speed, but for survival. But TP53 is only part of the story. Giant tortoises also have strong antioxidant defenses and mitochondria that produce far fewer toxic byproducts, protecting their cells for decades. They also possess low inflammation gene variants, preventing the chronic inflammatory damage that accelerates aging in most mammals. Their immune systems are unusually resilient, and their cells eliminate damage quickly, stopping tumors before they begin. Many of these traits likely evolved on isolated islands, where few predators and harsh conditions favored creatures that lived slowly and lived long. In tortoises, aging does not rush, it drifts quietly in the background of a life measured in centuries. In the far northern world of Greenland, where the ocean remains locked in ice and daylight fades into a permanent blue dusk, dwells a creature that moves as slowly as time itself. A silent giant drifting through sub-zero darkness, a being so old that it may have begun its life before the first European colonies in the Americas, before the invention of the telescope, before many human nations even existed. This is the Greenland shark, the longest-lived vertebrate ever discovered. Its lifespan is estimated at 250 to 500 years, making it the closest thing on Earth to an animal that lives across centuries as if they were mere seasons. But its longevity is not a mystery of myth. It is a triumph of biology. Scientists believe the Greenland shark's astonishing lifespan rests on three remarkable biological pillars. In waters hovering just above freezing, every chemical reaction inside the shark slows dramatically. Its heart beats slower than almost any known animal. Its cells divide at a fraction of the rate seen in warm-blooded species. Every metabolic process, from energy production to tissue repair, moves in slow motion. This extreme metabolic conservation means far fewer reactive oxygen molecules, far less cellular stress, and extremely slow accumulation of biological damage. Where mammals may age visibly in decades, the Greenland shark ages almost imperceptibly. Cold water becomes a shelter from time itself. To estimate its age, scientists turn to its eyes, specifically the core of the eye lens, made of proteins that form before birth and are never replaced again. These proteins hold radiocarbon signatures from the environment at the time the shark was born. They act as frozen timestamps, preserved through centuries of Arctic stillness. Through these molecular records, researchers discovered individuals that were alive during the age of sailing empires, alive during the signing of the Magna Carta, alive when Leonardo da Vinci walked the earth. Some Greenland sharks alive today predate the existence of modern science itself. The Greenland shark grows only 1 centimeter per year, one of the slowest growth rates ever recorded. This glacial pace means fewer rounds of DNA replication, fewer opportunities for mutations, and extraordinary genetic stability across hundreds of years. Their tissues contain high levels of TMAO, a molecule that protects cells from extreme pressure and cold, stabilizing proteins and preventing damage over incredibly long timescales. They also exhibit unusually low levels of age-related inflammation, a major driver of aging in mammals. And their metabolism is so slow that they require minimal food, often scavenging carcasses that sink to the sea floor, a lifestyle that further reduces biological wear. In the eternal twilight of the Arctic deep, the Greenland shark drifts through centuries with unhurried grace. Human civilizations rise and fall. Generations are born and lost, yet the shark continues its silent passage, aging so slowly that time itself seems reluctant to touch it, a creature for whom centuries are gentle ripples in an endless frozen sea. The naked mole rat is one of the only mammals known to display negligible senescence, a biological state in which aging itself appears almost absent. Unlike most mammals, it does not grow frail with time. Its risk of death does not rise with age. Decades pass, and its body barely changes. Hairless, nearly blind, and living in total darkness, this small rodent may look fragile, but beneath the surface lies one of the most durable biological systems ever evolved. In most mammals, aging follows a predictable curve. Cells accumulate damage, proteins misfold, cancer risk rises, and tissues slowly fail. In naked mole rats, this curve is almost flat. Scientists believe their resistance to aging is built on multiple overlapping defenses, systems that protect cells, tissues, and genetic material with extraordinary precision. Cancer is one of the leading causes of death in mammals. In naked mole rats, it is virtually unknown. Their cells produce exceptionally high levels of high molecular weight, hyaluronan, a dense, viscous sugar molecule that fills the space between cells. This molecule creates early contact inhibition, a mechanism that prevents cells from crowding and stops uncontrolled division long before tumors can form. It is a biological safeguard so effective that cancer almost never gains a foothold. Their genetic maintenance systems are equally remarkable. Naked mole rats show enhanced activity in base excision repair and nucleotide excision repair, molecular pathways that detect and remove DNA damage. Errors that would persist and accumulate in other mammals are rapidly corrected, preserving genome integrity across decades of life. Naked mole rats maintain a naturally low body temperature and an exceptionally slow metabolism. This reduces oxidative stress, slows chemical reactions that damage cells, and decreases the overall pace of biological wear. In essence, their bodies operate in a permanent low-power mode, aging slowed by design. Their underground world is harsh, low oxygen, high carbon dioxide, crowded tunnels, and scarce food. Yet naked mole rats thrive. They can survive severe hypoxia, switching to alternative metabolic pathways that would kill most mammals within minutes. These adaptations further reduce cellular stress and reinforce their resistance to aging. Despite their small size, naked mole rats routinely live 30 years or more, nearly 10 times longer than other rodents of similar mass. They remain fertile, active, and socially functional until the very end of life. In perpetual darkness, beneath the earth, a mammal has evolved not speed, not strength, but endurance. The naked mole rat does not outrun aging. It dismantles it, layer by layer, through biology engineered for survival. Over time, for centuries, scientists have searched for a way to defeat aging, to repair damaged cells, to restore youth, to extend life beyond its natural limits. But immortality, if it exists at all, comes with a price, because life is built on cycles, growth, maturity, and decline. If most animals age, and some resist aging, one jellyfish appears to reverse it. Turritopsis dohrnii, known as the immortal jellyfish, is the only known animal capable of repeatedly cycling between maturity and youth, not metaphorically, biologically. In the late 1980s, at a conference of marine biologists, a paper was presented that seemed impossible. Two laboratory students had collected what they believed to be a common hydrozoan species. They placed the immature medusae into a tank, hoping to breed them, and forgot about them. When they returned, they expected adults. Instead, they found fewer medusae, and newly formed polyps settled at the bottom. The adults had not reproduced. They had reversed. The jellyfish had transformed back into its juvenile form. Cells from the adult body reorganized themselves into an earlier stage of life. What the researchers had discovered was not Turritopsis nutricula, but a different species, Turritopsis dohrnii, and it was capable of something inconceivable. Through a process called transdifferentiation, one type of specialized cell becomes another. Directly. No stem cell resets, no intermediate stage. Damaged or aging tissues revert to earlier forms, effectively resetting the organism. When stressed by starvation, injury, or environmental shock, the jellyfish retracts its tentacles, shrinks into a cyst-like structure, settles onto the ocean floor, and begins again. Within days, a new polyp grows, and from that polyp, a new medusa emerges. The cycle can repeat indefinitely. Like other hydrozoans, its life begins as a planular larva, floating freely before attaching to a surface. It grows into a polyp, which clones itself into stacked juvenile medusae. Most jellyfish stop there. They mature, spawn, and die. But Turritopsis dohrnii has found a way around the inconvenience of death. In its cyst stage, the jellyfish appears to activate genes associated with DNA repair and telomere maintenance. Telomeres, the protective caps at the ends of chromosomes, normally shorten with each cell division. When they erode completely, cells enter senescence. In humans, this process defines aging. But in regenerating Turritopsis dohrnii, genes promoting telomerase activity are highly expressed, potentially restoring those protective caps and preventing cellular decline. It devotes energy not to growth, but to repair, not to reproduction, but to reset. This is not immortality in the human sense. It can still be eaten, still be destroyed. But absent predation or catastrophic injury, it possesses no biological expiration date, a loop, a system without a final chapter. Scientists now study this process for clues to cellular reprogramming. Could transdifferentiation one day help repair damaged tissues? Could understanding its telomere maintenance slow degenerative disease? Perhaps. But evolution separated us from jellyfish hundreds of millions of years ago. Some mechanisms may never translate. Originally native to the Mediterranean, immortal jellyfish now appears across the world's oceans, likely hitchhiking in ballast water. In laboratory conditions, individuals have been observed reversing their life cycle multiple times, some regenerating 10 times within months. In the wild, the cycle could continue far longer, possibly forever. In a world obsessed with youth, with longevity, with defeating time, a transparent jellyfish has quietly solved the problem in its own alien way, not by extending adulthood, but by refusing to remain in it. Immortality, it seems, is not about living forward forever. It is about knowing how to begin again. Long life is never given freely. It is negotiated through compromise. The tortoise trades speed for stability. The Greenland shark surrenders warmth for centuries. The naked mole rat accepts darkness for resilience. And the immortal jellyfish abandons permanence to begin again. Each species pays its price differently. Slower growth, lower metabolism, isolation, simplicity. Evolution does not hand out longevity as a gift. It demands exchange. But in these extraordinary creatures, we witness something remarkable. Life pushing against time, not conquering it, but reshaping it, bending it, slowing it, and in rare, fragile cases, looping back to the start. Perhaps immortality is not about living forever. Perhaps it is about learning how to endure.